So we're going to move ahead to the next speaker, Saroj Poudel. Saroj is another former ENIGMA postdoc. He graduated did his PhD with Eric Boyd at Montana State in Bozeman, where he was using phylogenetic methods to study the evolution of nitrogenase and other oxidative reductases. And then when he came to Rutgers, he was an EOAS and then later a NASA postdoctoral fellow, and finding his computational expertise and learned some experimental skills while he was in the lab to study the evolutionary process of early evolving proteins and peptides. And as of January, he's moved to a private company, Quantum SI, where he's working on developing technologies for next generation protein sequencing. So Saroj is going to talk to us today about the ecological distribution of nitrogenase reveals modern isoforms evolved to potentially bind charged proteins to mitigate oxygen toxicity.
Thank you, Vic, for the introduction, and thank you for having me here. Yeah, so I'm going to talk about my work that I did in the last 4 or 5 months while I was at Rutgers. And I'm just going to go over a few results. I was in the process of writing a paper. Hopefully this will be done pretty soon. But anyway, so I'm going to talk about an enzyme that's involved in nitrogen fixation. So I've been working on nitrogenase for almost 6 to 7 years, actually. I actually started on this enzyme when I started my PhD, and then have been looking into this since then. I want to start with going back in history and talking about why nitrogen fixation and what can we look into this enzyme so that we can better understand how it's functioning and how it's functioning and why it's diversifying into the environment. So on the x-axis, what you're seeing here is geological time scale. And on the y-axis, at the bottom panel is the partial of oxygen, basically showing how much oxygen was available at a particular time. And on the top panel is different metabolism that Eli plotted. And I'm going to draw your attention to nitrogen fixation, which is shown by the blue dotted line. So what you can see is based on the evidence, what we can say is nitrogen fixation was around roughly 3.2 billion years ago, right? And the lighter bar plot represents that we're not that confident, but we hypothesize that nitrogen fixation was before at least around 3.5 billion years ago. And so you guys have been seeing this plot many times, so I'm not going to go into detail. But again, so what this suggests is that nitrogen fixation existed before oxygen was around, right? So then the enzymes that function in these fixing nitrogen must or had an anaerobic origin. So these are potentially oxygen sensitive enzymes, which is the case for the modern nitrogenase as well. Looking at the nitrogenase itself, it is 3 subunit enzyme. We have H, D, and K. So H is that contains the fragments of a cluster. It's a homodimer. And D and K is the 2 big subunits that contains the modified fragments of a cluster, also known as P cluster, and the catalytic center, which we refer to as FeMO cofactor, which basically means that it has iron and molybdenum as its active site. And for this one, it's using ferredoxin as an electron donor. It's reducing ferredoxin to fix nitrogen to ammonia. Now, we all know that nitrogen fixation is a really expensive process, right? If you look at the equations, it roughly takes around 8 electrons and 16 ATP. So that's, for 1 electron transfer, it's 2 ATP. So that's a very expensive process. And remember in my previous slide, I showed that nitrogen fixation existed around 3.2 billion years ago. So it had billions of years to improve, and perhaps it was worse back in the days, but even the modern nitrogenase is a very expensive process. So what I wanted to understand was, why is it really requiring a lot of energy to fix nitrogen? And do we have an alternative options? And can we actually engineer nitrogenase to a better and efficient enzyme. So again, a few information on nitrogenase. So there are different ways, 3 ways to fix nitrogen currently. And the only way to fix nitrogen biologically is using the nitrogenase enzyme. Right? And that contributes to roughly 60% of the total reduced nitrogen. And it's also an oxygen-sensitive enzyme, which I showed in the last 2 slides. And I guess it's not surprising that it's immersed in an anaerobic organism. And in this case, we believe that it's a mechanism. But then it has since diversified into different metabolic backgrounds, aerobes, phototrophs, and so on. This was the work that I did in my last paper. Please follow that for more information on that. So nitrogenase, we can classify nitrogen into 3 different groups based on the medical factor. NIF, which has molybdenum and iron. Vanadium, which has vanadium and iron as the active center. And ANF, iron. So what I'm showing here is a phylogenetic tree, and you can distinctly see these 3 groups, NIF, ANF, and BNF. And NIF can be further divided into 4 subgroups, A, B, C, and D. And then again, you can see how distinctly they can group, right? And what I've done is overlaid with their oxygen requirement. So the organisms that encode these NIF isoforms are the aerobes or anaerobes and facultative. And it looks like NIF-A is mostly encoded by aerobes and anaerobes and few facultative. And the rest of the nitrogen they're mostly encoded by anaerobes or facultative anaerobes, right? So what this shows is somehow NIF-A has diversified into organisms that could survive in presence of oxygen. And again, remember, these enzymes, or at least the ancestral nitrogen, is immersed in an anaerobic settings. And beside NIF-A, most of the enzymes have stayed in anaerobic settings. And so this raises a question as to how NIF-A is able to function in an oxygen environment. Looking at the taxonomic distribution itself, it's widely found in bacteria and also found in phylum and eukarya. Remember, I mentioned that nitrogen is likely immersed in a mechanism, right? That belongs to Euarchaeotophyla. So it's not surprising that we see in archaea, but even though it immersed in archaea, it's actually widely distributed in bacteria, and it's not found in eukarya. So what I wanted to do was go into the environment and look at where they are found. So I collected metagenomes, only environmental metagenomes, which accounted to roughly 3,200, and looked for nitrogenase in those environment. So the dot or the circle that you're seeing here is all the metagenomes that I scanned for the nitrogenase, and the colored ones are the ones that encoded these nitrogenases, and there are different gradients based on the abundance, with the lowest colored blue and then the highest colored yellow. So what we can see from here is it looks like nitrogenase is usually found around 45-degree latitude. But then the trend that we're observing for the last 5 to 7 years is we're starting to detect these nitrogenase enzymes in higher latitude as well, like in Antarctica, right? And this is what this study is corroborating as well, is we're starting to see nitrogenase not around those thermal springs or moderate temperature environment, but also in colder environment. Now that could be because we're advancing in technology, there's a change in environment, there could be many things that could be contributing to the detection of nitrogenase in these environments. But this is the trend that we've been seeing for the last 5 or 6 years, and that's what we see as well. Which is again verifying that nitrogenase has been either diversifying into the environment, or we're starting to detect these into unknown environments as well. Taking a further look into the metagenome itself, what I'm showing here is a deeper look into the metagenomes I showed you before. I've classified these into surface and subsurface. The surface is anything that's 1 meter above and anything below that would be subsurface. And I've further classified them into terrestrial, which is colored as brown in the plot, and then the blue would be aquatic system, and then the same for subsurface. And I further categorized them into soil, water, and sediments, and then also gone further into what type of soil and what type of water, for example, pond, lake, or watersheds, and so on. So now this is a busy figure, and I'm not going to go in detail, but what I'm going to say is, briefly looking at this, what you can see is on the surface, there appears that nitrogenase has been diversifying into a different environment, but in subsurface, not so much. But what you can see is it's enriched in subsurface environment than in surface environment, right? So how can we know that? If you look at the gray bar plot in here, so that's the size of the metagenomes, and the colored ones are the nitrogenase isoforms. And you can see that in subsurface, they're not as enriched as in subsurface environment, right? So again, it's not really surprising because these enzymes have MERS in anaerobic settings. They don't like oxygen, so they would rather be in oxygen deprived or really reducing environments. Now we can't say that just because it's in surface doesn't necessarily mean that it's anoxic, but more or less when we classify into subsurface, they are strictly anaerobic for the most part. And again, remember, NIF-A has been diversifying into aerobic organisms as well, and which is what we see is both NIF-A and NIF-B are the ones that are highly abundant compared to NIF-C, D, and other isoforms. And in fact, I didn't really find a lot of NIF. That could be because these enzymes are, those isoforms are not widely found, or the environment that I selected just didn't have those nitrogenases. So what I wanted to look was, now that I have these new novel nitrogenases, I wanted to see if they are found, if new phyla has actually picked up these nitrogenases or not. Then we already know. I had already studied nitrogenases in different bacterial species in my last study. So basically I wanted to look at, can I find new phylum, right? And in fact, I do see NIF-A being picked up by firmicutes, Elucimicrobia, which was detected in soil environment, and multiple phyla in picking up NIF-B. So what this is suggesting that more NIF-B has been diversifying, or that more phylum has been picking up NIF-B than NIF-A, at least based on the study. Right now, this could be again because of sampling bias. So we couldn't detect nitrogenase before, and then now we're starting to see more nitrogenase. Maybe that could be the reason that I'm seeing more NIF-B being picked up by different phyla and in different environment, more so than NIF-A. So now that's interesting, right? So NIF-A and B being the most abundant isoforms, I wanted to understand how they've evolved over time. So I reconstructed a phylogenetic tree, and what I'm showing here is basically the distance. So when you start from a root, and I'm calculating the distance for each homologs, and I'm plotting, making a box plot, basically showing you the distribution of these homologs. And what that corresponds to is how much they have diversified since their ancestral nitrogenase emerged, right? So when you look at ANF, they've only diversified. So that's what the y-axis is. Cophonetic distance is basically a distance. Looking at ANF, it hasn't really diversified as much from their ancestral nitrogenase, and the same for BNF. And I've divided this into metagenomes and complete genomes. So the complete genomes data is from my last study, and then metagenomes is from this study. So so ANF and BNF, they haven't really diversified as much. And when you look at NIF isoforms, NIF-D, they have started to diversify, but not as much as NIF-A and B, right? So they're really diversified from their ancestral roots. And that's interesting. And in a way, I would say it's expected because, again, if you remember the phylogenetic tree that I showed you before, majority of aerobes are encode NIF-A as opposed to NIF-D and NIF-C, or ANF and VNF for that matter, right? So I wanted to understand what's happening in NIF-A and NIF-B that they're diversifying into compared to the ancestral roots, and how they're able to survive the oxygen toxicity. So I relied on statistical coupling analysis. So that approach is using Kullback-Leibler divergence. It's basically an approach to score conservation. So you can look at each positions and identify evolutionarily conserved positions. So the bottom panel is NIF-B. And again, remember, nitrogenase has 3 subunits. So the one that I'm showing you here is only 8 subunit, but I did for both D and K as well. So the bottom panel is NIF-B, 8 subunit, and on the top panel, I'm taking the difference between NIF-A and then NIF-AB. So what that means is I'm trying to highlight the positions where NIF-B is diverging away from NIF-B. And I can put what I've done is put a threshold. So for example, if you look at the bottom panel, I have a red dot at 2.5. So anything above that, I'm saying that these positions are evolutionarily conserved, right? And I can look at each positions as shown in the y-axis, x-axis, right? So so you see multiple peaks in NIF-B. So what that is, again, is based on this approach, it thinks that those positions are really conserved. And what I've done is pointed out few positions with red dots. So what that is if you look at the top panel, I'm taking the difference between NIF-A and NIF-B because I wanted to look at how has NIF-A diversified from NIF-B. So to do that, again, using the score, I can look at positions, and I put a threshold of 0.25 in this case. And there are several positions where NIF-A has diversified and are really conserved. And then you can see that there are a few positions that I put a lower threshold as well. So any peaks that you see going downwards, that basically means that NIF-B is selecting for those positions compared to NIF-A, meaning that for NIF-A, that position is not really relevant, either for function or structure, but NIF-B really needs those positions, right? In which I'm highlighting in as red dots in there. So NIF-B really has 4 or 5 positions that it's selecting compared to NIF-A. And when I took these positions and plotted on a structure, what I observed is that in the 8th subunit, which is showing you by the red dot, red circle here, 8th subunit is a homodimer. And what I observe is they have this pocket, which is really charged, right? Shown by different blue and red and white. So red is negatively charged, blue is positively charged, and white is neutral charged, right? So you can see that that pocket is really charged compared to the other subunits where I see a distribution of the charges, but not so much so in this pocket. And the reason, and that's the case for NIF-A and B. When I also look at NIF-A versus A and F, and I see a similar pattern where NIF-A, when compared to NIF-B, I see this charged pocket. So how is this really helping NIF-A compared to NIF-B or or A and F? So a recent study, they found this protein called Shethna. It's basically a 2-ion supercluster protein. It's always found in dimer form. It's a homodimer. And what the paper predicts is that nitrogenases are binding to this protein, and it's allowing nitrogenases to be stable in an oxygen, in high oxygen condition, not only for a certain time. So NIF-A by itself can survive to roughly around 10 to 20 minutes of oxygen exposure. I think they did with 2% oxygen exposure, but in presence of this protein, I think it can go a little bit longer than that. Now, we don't know how it's protecting this oxygen, but the prediction is that when the nitrogenase binds to Shethna protein, this deactivated nitrogenase, it protects from this oxygen, and when it's active, the Shethna proteins leave the binding site, and then it can be active. Now, again, that's just a hypothesis. We don't really know how it works. But again, so what this is showing is that NIF-A having this charged pocket is an indication that they're moving towards more, they're moving towards where they can function, where they can be stabilized in an oxygen environment, and by binding to proteins as Shethna proteins, right? Compared to NIF B or C or D for that matter, and which could explain why they're found in aerobes or other metabolic backgrounds. So with that, what I've shown today is nitrogenase, since its emergence, it has 5 more isoforms, ANF, VNF, and then A, B, C, D for NIF itself, right? And even though it emerged really early in anaerobic settings, it has diversified into aerobes and other metabolic backgrounds. And I also showed that not only it's only found in a moderate temperature environment, but we have been starting to see in really cold temperature. And the taxonomic distribution that I showed indicates that we can see nitrogenase in Neophilum as well. And lastly, looking at NIF-A, the structural comparisons in NIF-A and NIF-B indicates that NIF-A has evolved into including these binding, including the charged pocket where it could bind these proteins, such as Shethna protein, that it could help them stabilize in presence of oxygen. Yep, that's all I have. With that, I'd like to thank my lab and everyone else involved in this project. And I'm happy to take any questions.
All right, thank you, Saroj. Yeah, so Akif is asking whether or not the Shethna protein is expressed predominantly during dormant phases when the organism is not doing nitrogen fixation. Is that fair?
Yeah, I think that's what the prediction is, yes.
Saroj, do you know if the Shethna protein shows up in a lot of the same genomes where you have NIF-A, where you have predominantly NIF-A? Is there any correlation with genomic occurrence of those isoforms of nitrogenase in this particular protein?
I haven't looked into that, but that would be interesting. Yeah, it would be interesting to see if that's the case. And if NIF-A is actually using Shethna, I would expect it to correlate. But yeah, I haven't looked into that yet.